This video is on organelles and their features. So, what we really want to get out of this video today, two main things. For each part, we want, first of all, the structure of the organelle, and then secondly, the function of it. Those are the two things we're looking for in each of them. So, structure, what does it look like? What's it made out of? Function, what does it do? First one here, the plasma membrane. Okay, it's made out of phospholipids, so that's the structure. Okay, we've seen phospholipids before. Here they're going to form something called a phospholipid bilayer, where we have the hydrophobic tails oriented towards each other and the hydrophilic heads oriented towards the liquid or water side. And so it forms this bilayer that we can see down here. The cool thing is this happens just naturally in water. So if you have a bunch of phospholipids in water, they will naturally form a phospholipid bilayer. Looks like that. Function, well, it's to provide a boundary between the cell and the outside world. It also allows stuff to come in and out of the cell. It also serves the plasma membrane, serves to give both that barrier to the inside world and the outside world. And there's also membranes within the cell made out of phospholipids too. Okay, so where are these? Well, these are going to be around the outside of the cell, the plasma membrane. And anything within the cell is also going to be made out of this phospholipid bilayer. We're not going to call it the plasma membrane, it's just going to be a membrane. But anything that's got a membrane within the cell is going to be made out of this phospholipid bilayer. Cell membranes often contain proteins embedded within the phospholipid bilayer. So the plasma membrane is not 100% just straight phospholipids. There's other stuff embedded in it. They have some surface markers that identify what kind of a cell it is. So your cells might have markers to indicate that it is, say, a skin cell versus a red blood cell versus a nerve cell versus a muscle cell. There are proteins on the surface that are embedded in the plasma membrane identifying that particular cell within your body. There's things like cholesterol embedded within the membrane, which helps give it some structure and support. We have things like transport proteins, which allow stuff to come in and out of the cell. And those are going to be regulated by certain things within the cell. We have receptor proteins, as recognizes and binds to the substances outside of the cell. So there's all kinds of stuff embedded into the membrane beyond just phospholipids. The plasma membrane is surprisingly dynamic, meaning it's constantly in motion. It's not everything's just locked into one place. It's kind of like, think of it sort of as like things floating on an ocean, boats floating on an ocean. It's able to move around. We call this the fluid mosaic model. It states that the phospholipid bilayer behaves like a fluid more than it behaves like a solid. It's not really like a solid barrier. It's more like a liquid barrier. It's very fluid. Proteins and lipids can move laterally through the bilayer like a boat on the ocean. It's kind of cool. So they're, again, not locked into position. They are able to move around. This results in a mosaic, so it's a constantly changing membrane. The big term here being the plasma membrane forms a fluid mosaic model. Okay, so the nucleus. This is not going to be in every cell, but many cells. The function here is to direct the cell's activities and store DNA. It is filled with nucleoplasm, so in terms of structure, which is a jelly-like liquid. Before cell division, the chromatin, which are going to contain all of your DNA, are going to condense into chromosomes. And it's surrounded by a double membrane called the nuclear envelope. And guess what? It's another membrane. It's going to be made out of, phospholipids. Okay? It's called the nuclear envelope. It's made of two phospholipid bilayers. What that allows is for certain things to enter or leave the cell. I know this looks like it got blocked out here by the picture, but essentially those openings there in the double membrane allow things to enter and leave the nucleus. Okay, so through those openings, they are nuclear pores. The nucleolus, the center here, okay, it's much more dense. When you're looking at it through a microscope, you'll be able to tell it is a much denser, darker part of the nucleus. This is DNA that is concentrated when it is in the process of making ribosomal RNA. So the nucleolus is going to be responsible for making ribosomal RNA, which is going to be seen as rRNA, little r RNA. This is responsible for making ribosomes. So the genetic code that is responsible for making ribosomes is going to be inside of this nucleolus. The ribosomes, these are organelles made of protein and RNA that direct protein synthesis in the cytoplasm. These are very tiny, okay? Very, very small relative to other features here. Basically, when you're looking at them under microscope, they're just going to look like little dots at the best resolution. They are very, very tiny. But the nucleolus is responsible for making ribosomes. Ribosomes are going to be responsible for making proteins out in the cytoplasm, so outside of the nucleus. Okay, another feature is called mitochondria. All right, so it's going to kind of look like this. They kind of look like, I don't know, jelly beans or kidneys or something like that if you want to liken it to something. The function is to harvest energy from organic compounds and transfer it to ATP, which you might remember from the last chapter as adenosine triphosphate. It is the energy currency within the cells, energy currency. So it's how they pay for things. It's how they make reactions happen. They pay for it with ATP. It's made in this structure called the mitochondria. And check it out. It's got a membrane here. It's got a lot of membrane. It's got a whole bunch of surface area. And that membrane is, yes, you guessed it, made out of phospholipids. Okay. So, a lot of surface area here with these folds. Okay, those folds are called cristae. You have an inner membrane and outer membrane. It's all made out of phospholipids. So that's the structure of it. The function is to get energy from organic compounds and transfer it to ATP so the cell can pay for reactions. Okay, so structure-wise, said it on the last slide, but here you go. ATP powers most chemical reactions in cells. Active cells have lots of mitochondria. So since this stuff makes energy, cells that are doing a lot of stuff are going to have lots of mitochondria. So for instance, if you look at muscle cells, we should see more mitochondria than, say, a cell that doesn't need to do as much. Okay, like a skin cell, you're not going to need to do as much. You're not going to have as much mitochondria in it. Cells that are not very inactive, or cells that are very inactive, do not have many. So like fat storage cells, not doing a whole lot, not going to have a lot of mitochondria. It has an inner and outer phospholipid membrane. Folds on the inside are called cristae. It contains proteins that carry out the energy harvesting reactions. So that's where the energy is actually made. The ATP, okay, is getting energy here along these cristae. Mitochondria actually have their own DNA, which is kind of an interesting side note here, separate from the nucleus. It can be reproduced by division of pre-existing mitochondria. And so the fact that it has its own DNA has some people believing, some scientists believing, that it actually originated from a prokaryotic cell that were incorporated into eukaryotic cells. So you've read about prokaryotic cells so far. Not eukaryotic yet, but the idea being that these simple celled organisms, prokaryote, somehow became incorporated into eukaryotes early on, an example of symbiosis. This is two organisms using each other to survive. So that's kind of an interesting theory there. This provides a safe place for prokaryotes and provides energy for eukaryotes. So there's a mutually beneficial relationship there. Each has something to gain. Okay, ribosomes. We said a little bit ago. Okay, those are being coded for by rRNA. rRNA, and that is found in the Nucleolus. Okay, these are small spherical organelles that are responsible for building proteins. As I was saying earlier, really these things are just going to appear as little dots. Even at the highest resolution, you're not going to see these really, really well. Okay, but they are responsible for building proteins. They don't have a membrane. They have a large subunit and a small subunit. And they are just sort of free in the, some of them are free in the cytoplasm or cytosol. Some are attached to the rough endoplasmic reticulum, which we'll talk about here in just a moment. But their job is to make and put together proteins. Okay, so these things are putting together peptide bonds or linking amino acids together, right? We've talked about these terms in the last chapter. Those are all being done with ribosomes. Okay, the endoplasmic reticulum. It's a system of membranous tubes and sacs called cisternae. It functions like an intracellular highway. So this is kind of the getting stuff to where it needs to go. It moves molecules from one part of the cell to another. So it's kind of like the shipping department. Okay, it moves stuff around. It's made out of membranes. So yes, once again, you guessed it. This is made out of phospholipids. Okay, so that's what it's made out of. Structure and function, it moves stuff around. It's the shipping department. There's rough and smooth types of endoplasmic reticulum. Okay, so here's kind of what they look like in terms of the picture from the cell. So it's kind of just these flattened sacs. Kind of this one, in this case, they're close to the nucleus there. Sort of flattened sacs that look like that. Right inside of those folds there are what are called cisternae, kind of like the cristae in the mitochondria we saw just a minute ago. And then if you have rough endoplasmic reticulum, you notice there are little dots all over this thing. Guess what those dots are? Those are ribosomes. Okay, so rough endoplasmic reticulum is lined also, in addition to being made out of phospholipids, it's lined with ribosomes. Okay, and the smooth endoplasmic reticulum that doesn't have the dots on it will not have ribosomes. Okay, so rough ER, we kind of already said a little bit of this. The function is to prepare proteins for export or insertion into the cell membrane. So we're making proteins and getting them ready to be used correctly. It contains ribosomes, which we've said. It produces phospholipids and proteins. So it's making a couple things, not just proteins, but also phospholipids. Proteins are then exported from the cell or inserted into the cell membrane, so they go to where they are supposed to go and be used. Ribosomes on rough endoplasmic reticulum make digestive enzymes. That means it's going to break stuff down. Okay, and probably something that's going to perform hydrolysis reactions that we talked about in the last chapter. These accumulate inside the rough endoplasmic reticulum, little sacs or vesicles. Okay, so basically little membranes, little pieces of membrane, then pinch off and store enzymes until released. These are most commonly found in cells that produce large amounts of protein for export. Okay, cells in the digestive tract, things like that. Smooth endoplasmic reticulum. So this one's going to not have ribosomes. It lacks ribosomes and appears smooth, so there's no dots all over it. Most cells contain very little smooth endoplasmic reticulum. Don't usually have a whole lot of it. The function is to build lipids, in particular steroids, things like cholesterol. The function also is to produce estrogen and testosterone, okay, things that are also steroids. Right? They're abundant in liver and kidney cells. That's where you're going to see them the most. There, in particular, their job is to help detox drugs and poisons. So things that go into your system that your body doesn't like, that it needs to metabolize and get rid of. Okay, you're going to have lots of smooth endoplasmic reticulum in those particular locations, liver and kidneys. What ends up happening if you are constantly having to detox these drugs out of your system, so if you were like abusing drugs and alcohol, those cells produce more smooth endoplasmic reticulum to help combat that high intake of toxins. And over time, what that leads to is drug tolerance. So you need to take more of the drug to feel it, and that's because you've built up a whole bunch of smooth ER, okay, which is going to simply just keep metabolizing it faster and faster, and so you need more of the drug to get the same effects. That's a real problem there, but if you look at the cells, like the liver or kidney cells of somebody who's an alcoholic or drug addict, you're going to see a lot more smooth endoplasmic reticulum in them than the average person, and that's because they've built up tolerance to these drugs. Smooth and rough ER form an interconnected network. It's all kind of a flattened membrane, okay? They all kind of look like flattened sacs on top of each other, okay, is what you're looking for there in terms of what the smooth and rough ER look like. The rough will have ribosomes on it. The smooth will not. Okay, the Golgi apparatus. Okay, this one, it's also a flat membranous sac here. The function is to modify proteins and give them address labels. Okay, so this one is like the actual UPS. The last one sort of got things ready to go. This one's actually, here's where it's going. Flat membranous sacs. The sacs nearest the nucleus received vesicle. So here you can kind of see the nucleus sending out, over here in our picture on the right, sending out a vesicle or a small membrane sac, okay, towards this Golgi that they've dyed purple. Okay. And it moves away from the nucleus to get modification. So as it goes through the Golgi apparatus, it gets shipping labels put on it, told where to go, it gets adjusted a little bit, okay? And it's going to ship it to the location. Vesicles, so we've kind of already said this, but what are they? They are small spherically shaped sacs that are surrounded by a single membrane and that are classified by their contents. Okay, so they're membranes, right? So they're generally spherically shaped, okay? And on the inside is some kind of protein or something that does a whole bunch of stuff, okay, just waiting to either be shipped or moved or opened up and released. They often migrate through the cell and fuse with the plasma membrane and release their contents out of the cell. So what you can imagine here, if this is an entire cell, and here is a vesicle, okay, it moves this way, and eventually it fuses with the membrane and spews all of its contents out of the cell. There's different types here. We have things like, for instance, lysosomes. These emerge from the Golgi and contain digestive enzymes, so they might break up inside of the cell. Sometimes in our drawing, like I did up here, it'll merge with the plasma membrane and release the contents. Sometimes, okay, they may come apart within the cell and release their contents inside of the cell. Okay, so we can do either one there depending on what's inside of this vesicle. Some other functions, they can break down large molecules like proteins, these lysosomes, I should say. Other functions can break down old organelles, something called autophagy. Another function, program cell death. So let's say the cell is old and it's no longer helpful. In fact, it can become a liability. It can go through something called apoptosis, where the enzymes inside of there just destroy the entire cell. Kind of a programmed self-ending lysosome. Okay, another example here is something called peroxisomes. They're similar to lysosomes, but they contain different enzymes. These are not made in the Golgi. They're made elsewhere. They're abundant in kidney and liver cells, so you might guess, based on what we saw earlier about people with lots of endoplasmic reticulum and things with kidney and liver cells, these are to help break down toxins. The function is to neutralize free radicals, which are dangerous molecules. It's named for hydrogen peroxide with the formula H2O2 that's produced when breaking down alcohol. The function here is also to break down fatty acids in addition to breaking down alcohols, and the mitochondria use those fatty acids for energy. Okay, so kind of breaking all this stuff down that we've talked about. There's been a lot here. We start with talking about the nucleus and then what's going on in the nucleolus. We talked about the different types of endoplasmic reticulum. We talked about the mitochondria. We've talked about the Golgi. We've talked about ribosomes. So how does this all go together? So how do cells, this is an important point here, make proteins? All right, so step one. We've kind of talked through this, but we're going to go through it all together here. Step one, the proteins are assembled by ribosomes on the rough ER. So it's going to be read here in the nucleus and then assembled out here on the rough endoplasmic reticulum. Okay. Step two, vesicles carry the proteins from the rough ER to the Golgi apparatus. So it passes through the endoplasmic reticulum, arrives at the Golgi. Step three, proteins are modified in the Golgi apparatus and enter new vesicles. So they enter a new container, a new membrane of sorts. Step four, some vesicles release their proteins outside the cell. So from the Golgi, they go to the edge of the cell, okay, and then they spew their contents outside of the cell. Other vesicles remain in the cell and become lysosomes and other vesicles. So this would stay inside, waiting to, say, release its contents back into the cell or to break something down. All right, so we have a couple of options for what those can do. Okay, so now we're going to take a look at the cytoskeleton. And kind of as the name suggests, and just like what you have a skeleton system that's going to support you. There are structures within the cell that aren't necessarily organelles, but they are helping the organelles stay in their current place. They kind of anchor stuff where it should go. It helps keep the nucleus in the center of the cell, helps keep the ER and the Golgi around it. So the cytoskeleton, it's a network of thin tubes and filaments that stretches throughout the cytosol. And when you're looking at stuff through a microscope, you're not really going to see it. It's all very tiny. You can only see it in certain situations, which we'll talk about later. The function, it gives strength and support to the cell's structure, like I said, just like your skeletal system. Another function acts as a path on which molecules move, so it kind of does act as a highway for things to travel along. There are different parts to this, just like your skeletal system isn't just your bones, right? You got other things going on there as well to help support you, muscles, filaments, things like that. So one of those here for cells are called microtubules. These are hollow tubes made of a protein called tubulin, right? That big ending there with IN telling you that it is, in fact, a protein. They radiate outward from centrosomes near the nucleus. Okay, so if this is the middle of the cell, right? Okay, these are going to go outwards. Okay, not necessarily all the way to the end. Maybe they branch out like that. Okay, but they are going outwards. It's kind of a network, all right, going through the entire cell. The function is to hold organelles in place, maintain cell shape, act as a track that guides organelles and molecules as they move inside the cell. So it kind of anchors things in the middle here, and maybe it holds the endoplasmic reticulum and the Golgi in place, okay, holds the mitochondria over here, all right, that's its job. This is called microtubules. Okay, so again, you're not going to be able to see it with necessarily just looking at a microscope unless it's a very specific set of conditions, but here's kind of what it is. They sort of act like sort of bracing stuff, okay? They anchor stuff in place. They hold it where it's supposed to be held. Okay, microfilaments, another type here. We have microtubules, now we have microfilaments. These are long threads of actin. Guess what? Another IN ending there. That is a protein. It's linked end to end and wrapped around each other like two strands of a rope. We'll see what this looks like later on this year. The function is to contribute to cell movement. You see a whole bunch of these things in especially muscle cells as they move back and forth and slide over each other. There's a whole bunch of actin that allows that to happen. It also happens with white blood cells. They kind of crawl along looking for certain things, but big part of muscle cells, actin. We have something called intermediate filaments. These are rods that anchor the nucleus and other organelles to their places within the cell. So that's their specific job, right? Holding it exactly where it's supposed to be. It maintains the internal shape of the nucleus. Okay, so here's kind of what those look like, all put together: microtubules, microfilaments, intermediate filaments. Okay, their structure and their function, all summarized together. Okay, cilia and flagella. You read about these when you were reading about prokaryotes. So these are generally, not specifically only two, but they are generally going to be found with bacteria. Okay, these are hair-like structures that extend from the surface of the cell. The function is to assist in movement. Okay, they allow the cells to move around. They're going to be made out of those microfilaments, microtubules, things like that. Cilia, these are short and present in large numbers. It helps detect vibrations such as sound. Okay, so like the cilia then, okay, are going to be a whole bunch of, they almost look like hairy structures coming around the outside of a cell. Okay, they can help it move. It's kind of like say like a fin with a fish, but they can also like detect vibrations, things like sound, okay, and respond accordingly. Flagella, these are longer and present in small numbers. Usually you only have one of them. Right? And a big example here would be like human sperm cells, but what these will look like, generally speaking, okay, if you have like a bacteria sort of shaped like that, okay, a flagella would be sort of a long whip-like tail that the cell can then move back and forth, so it can kind of go either direction here and help propel that particular cell in a different direction. Okay. The structure of these are going to be 9 pairs of microtubules around 2 central tubules. You'll see what that looks like here. Okay, it kind of looks like that. So if you were to break, here's a picture. This is a cilia there. Okay, on the outside of the bacteria. This might be a eukaryotic cell as well. They are found there as well. Now, if we look inside of it, okay, you have 2 there in the middle, surrounded by, I think we said 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yep. Okay, and that helps that cell move around. Okay, and then lastly, and we're going to get to these later on this year in more depth, are centrioles. Okay, these are 2 short cylinders of microtubules at right angles to each other. Okay, so if you can draw a cylinder, all right, they're going to be found next to each other but at right angles. They're situated in the cytoplasm, that's where they are, near the nuclear envelope. Animal cells have them, plant cells do not have them, right? So X on plant cells, but animal cells do have them. And what do? They are going to help the cell during cell division. They help it to divide evenly. So they pull on things using a lot of microtubules there, which you can see kind of in the green. They're pulling on, those are chromosomes that are being stretched, okay, into 2 new cells. One of these is going this way, one of these is going that way, pulling apart into 2 new cells. So there you have it.